So far, we have learned how to access and slice a DNA sequence. Today, let's solve a new problem. Suppose we want to read a DNA sequence in the opposite direction. How can we ask Python to do that? Here is our DNA sequence. If I asked you to reverse it, you could probably do it by looking at it. But what if this sequence contained thousands or even millions of nucleotides? Reversing it manually wouldn't be practical. So let's see how Python can help us. Notice something very important. We are not changing any nucleotides. The A is still A, T is still T, and so on. The only thing that we are changing is the direction in which we are reading the sequence. Earlier in this series, we learned that Python slicing has three parts: a starting index, an ending index, and a step. Today we'll use that knowledge to solve this problem. Today's lesson is really about understanding the step. Here's something to think about. What do you think will happen if the step becomes negative? Will Python still move forward through the string, or will it start moving backwards? Let's find out. First, let's store our DNA sequence in a variable called as DNA underscore sequence. Now look carefully at what's inside the square brackets. I have kept the starting index empty and I have also kept the ending index empty. That tells Python to consider the entire DNA sequence. The only thing that I have changed is the step, and I have made it -1. Before I run this code, take a moment and predict the output. Where do you think Python will begin reading the sequence? Interesting. Python didn't start with the first nucleotide, instead it started with the last nucleotide. And this is because the step is -1. You can imagine Python placing its finger on the last nucleotide, then it moves one position backwards, reads the next nucleotide, moves one position backwards again, and keep doing this until it reaches the beginning of the sequence. This picture shows exactly what Python is doing internally. It is simply walking through the DNA sequence from right to left, one nucleotide at a time. Now let's connect everything together. Since the starting index is empty, Python knows that it has to start reading the sequence from the beginning. And since the ending index is also empty, Python continues until it reaches the other end. And because the step is -1, Python moves backwards instead of forwards. Here is the biggest takeaway from today's lesson. Many beginners think that the step only tells Python how many positions to move, but that's only half the story. The step also tells Python which direction to move. A positive step moves forward and a negative step moves backward. Once you understand this idea, then this syntax becomes very easy to remember. So whenever you have to reverse a DNA sequence, you don't have to write loops or rearrange the nucleotides yourself. Just ask Python to go through the entire sequence with a step of -1, and Python will return the reversed DNA sequence for you.